back, everyone. Let's do some biochemistry flashcards together. So, what type of enzyme catalyzes redox reactions that involve the transfer of electrons? That would be, so we are talking about the transferring of electrons, not side chains. So, it's not a transferase. Now, catalyzing redox reactions, it should be an oxidoreductase. It's the opposite, right? It's oxidoreductase. Nice. So an example of oxidoreductase is alcohol dehydrogenase. So it is removing the hydrogen from alcohol and giving it to NAD plus that turns into NADH. And as a result, from alcohol, you get acetaldehyde. And acetaldehyde reminds me of an aldehyde that definitely has acetal, so we need to know all the acetal structures. So we can get to that in a moment. Okay, for this we can put a good, or actually let me go back and write one example. Enzymes are reusable catalysts that are not changed by the reactions they catalyze. They are unchanged, catalyze both the forward and reverse reactions. So an uncatalyzed reaction requires a higher activation energy than does a catalyzed reaction. So the catalyzation turns the reaction energy, the activation energy, it brings it lower, it brings it down. There is no difference in free energy between catalyzed and uncatalyzed reactions. The free energy, however, the difference, the delta G, is not different. What type of enzyme catalyzes cleavage with the addition of water? It should be a hydrolase. Yes, since we have water, it's a hydrolase. So the hydrolase does the cleaving. We have hydrolysis to denature proteins, to break down proteins. And here we have an example of phosphatase. So we want an example. So phosphatase cuts the phosphate group and kinase adds the phosphate group, right? Example. This amino acid. So these flashcards are from chapter 1 and 2 only of the biochemistry chapters. So chapter 1 is about amino acids and proteins, and chapter 2 is about the enzymes. The amino acid is glutamate, polar negatively charged, aliphatic, acidic. So first of all, aliphatic means that it is polar, right? Or let's see, I forgot. Aliphatic side chains. Aliphatic refers to organic compounds that have open, straight chains or branch structures rather than cyclic structures. So yeah, aliphatic is the opposite of cyclic. There are three aliphatic amino acids: glycine, alanine, and valine. There's more because glutamate is aliphatic. So Let's see. We know glutamate has one more branch than aspartate, and this is acidic, so it's negatively charged. The basic ones with the NH3 plus, they are positively charged, and the side chain, the functional group, will be one carbon and then the next carbon, and from that carbon we have a carboxylic acid with the OH at the end. Yes. So this is a conjugation, I believe it's called. So this is where you get the double bond, and this is where you get a single bond attached to the OH. Cool, we're good for this. So I'll put it as, yeah, the good on this flashcard would reappear in 5 months, which is a long time, so that's fine for me. My exam is like in less than 3 months. So what type of enzyme catalyzes cleavage without the addition of water or the transfer of electrons? Cleavage without the addition of water. So you don't need to add water or the transfer of electrons. So when you don't need water or transferring any electrons to cleave the bond, it should be a lyase, right? Something like that. Lyase, yes. Catalyze lysis of a substrate, generating a double bond in a non-hydrolytic, non-oxidative elimination. I have no clue. I basically got lost after this part. So we have pyruvate, and then we have pyruvate decarboxylase, and it just removes a carbon dioxide, the CO2 from here, and just remains with acetaldehyde. So acetaldehyde actually has 2 carbons. So it catalyzes the cleavage without any addition of water or transferring of electrons. The transferring of electrons should be the oxidoreductase, right? So let's see. Here we have so we want an example, right? Which is pyruvate decarboxylase. Interestingly, this amino acid is glycine, so it has only one hydrogen and it's aliphatic, it's non-cyclical, and it's non-polar. I'm good with 5 months. The only amino acid is not chiral. Proline either breaks or kinks a helix, both because it cannot donate an amide hydrogen bond. That's about proline having no amide hydrogen, and also because its side chain interferes sterically with the backbone of the preceding turn. Inside a helix, this forces a bend of about 30 degrees in the helix's axis. 30 degrees, do we need to know that? Super specific, but anyways. Though the following visual is helpful to label methionine under polar. So methionine is not polar. Let's look at what was methionine, the M with the S on the second one, right? Second last, instead of carbon, you have S on second last atom. So What type of enzyme catalyzes the interconversion of isomers? It's isomerase. So we have phosphoglucose isomerase. It turns G6B into F6B. So instead of glucose, we have a fructose. And what is the difference? So far, here is no difference, but on the last two groups right here, you exchange the double bond of oxygen, and that makes it a F6B instead of G6B. So doing an example for this, phosphoglucose isomerase. But you need to know that enzyme, that's very important because it's part of glycolysis. Let me do that again. So phosphoglucose isomerase. What are clockwise coils? Since it's coil, it should be alpha helix around the central axis and are a common secondary protein structure. Alpha helices, coils, secondary structure. We're good with this. Just something here is the hydrogen bond that you see here, even between the nitrogen too, with the oxygen here from the carboxylic side of the protein amino acid. And here it's also you have the hydrogen sticking out, and inside they make so the hydrogen from the amino group forms bonds with the oxygen from the carboxylic group of the amino acid on the other side. That's why you get the beta pleated sheets and alpha helices. What type of enzyme joins two large biomolecules, often of the same type? So, type of enzyme joining two large biomolecules. That would be joining two large biomolecules, often of the same type. This should be a ligand, or I don't know. Ligase. I was close. I just needed the A's. So we have DNA backbone here. We have DNA insertion, DNA insert, stick in. You ligate them. You have a ligase that does that. Two large biomolecules like DNAs. You get a recombinant DNA. That's what happens. This is basically the microscopic view of what happens in genetic recombination. And we call this the recombinant DNA because recombination took place here and we had a crossover, recombining the DNA. You combine them together again, but from another homologous chromosome or sister chromatid, for example. So here we have ligase. This amino acid, let's take a look at it. This should be phenylalanine. You can see the phenyl here. It's phenylalanine, PHEF, and the characteristic of the side chain is that it is non-polar and it is not aliphatic, it's cyclic, right? And I think that's about it. So aromatic, non-polar, aromatic. And we have hydrophobic also as its characteristic. and aromatic molecules can absorb UV light. And what else? Yes. Cool. But do we see it here? Phenylalanine. Nice. We're good with this. So they can absorb UV light. This can be important since then they can absorb the UV light. How? Why? Due to the conjugation that takes place here. They have in their aromatic ring. I'll just put it. I might want to see this again, so yeah, just put it hard. What type of enzyme catalyzes the hydrolysis of fats? It should be a lipase. So we have lipase here. It uses water. So hydrolysis does take place here. Is lipase a subtype of hydrolase? Is lipase a subtype of hydrolase since it uses water to break down fats? Yes, lipase is a subtype of hydrolase. Hydrolases are a broad class of enzymes that catalyze the hydrolysis of various substrates by utilizing water to break down. Lipase specifically is a subgroup of hydrolase that targets the hydrolysis of lipids into their constituent fatty acids and glycerol molecules. The hydrolysis of fats by lipases involves the cleavage of ester bonds in triglycerides, resulting in the release of fatty acids and glycerol. This process requires the presence of water to break the ester bonds, and lipases facilitate this hydrolytic reaction. Lipases are crucial for the digestion and absorption of dietary fats in organisms as they enable the breakdown of complex lipids into more easily absorbable forms. Nice. We have a lipase here, as you see. It's breaking this carbon-carbon bond here, I believe. Am I right? Oh, it's cutting down the oxygen and carbon bond here, and the water, the H from the water attaches here that makes the OH, and apparently this carbon oxygen bond was enabling the triglyceride to be a fat. So we have tri of the glycerides. These are glycerol molecules, right? And they are cutting down the oxygen carbon bond. Apparently, we have the whole oxygen carbon bonds for fats. That's why they are fats. So a fatty acid is having the carboxylic acid, and then the fat is attached here with the, so it was the oxygen and then the carbon backbone. That's why actually carbohydrates give us fats, turn into fats, because they're just essentially the same. They have the same structure, atoms, carbon and oxygen. And yeah, and this is where you have a monoglyceride. So cool. What structure of proteins is the 3D shape? It's the tertiary structure of a single polypeptide chain. Stabilized by what? By hydrophobic interactions, such as acid-base, disulfide bridges bonds, hydrogen bonds. So these are hydrophobic interactions. We need to keep in mind that we have hydrophilics too. So hydrogen bonds are not, or are they? Let's see. Are hydrogen bonds, hydrophobic or philic? It should be philic, right? Or hydrophilic, meaning they have an affinity for water. They are bonded, they form between a hydrogen atom bonded to an electronegative atom, like oxygen and nitrogen. Does sulfur also bond to hydrogen? I'm sorry for that. So we have hydrogen sulfide, which is H2S. And it's a common example of compound where sulfur and hydrogen are bonded together. So it is a covalent bond. So hydrogen bonds are covalent bonds because they are sharing electrons. So one electron comes from the hydrogen and the other electron comes from the sulfur or oxygen, and they share their valence electrons, and these are covalent. But ionic bonds are different. Are ionic bonds covalent? And covalent bonds are two distinct types of chemical bonds. Ionic bonds are formed when electrons are transferred from one atom. One atom loses electrons to become a positive charged ion, and they call this the cation, and another atom gains electrons to become a negatively charged. The attraction between the oppositely charged ions results in an ionic bond. They are typically between metals and non-metals. So NaCl, for example, right? Covalent share, two atoms share one or more pairs of electrons to achieve a more stable electron configuration. Covalent bonds are typically formed between nonmetals. So when we have oxygen and hydrogen, right? So, and what are acid-base interactions? What are some examples of acid-base interactions in protein in tertiary proteins? In tertiary proteins, acid-base interactions typically involve the ionizable groups of amino acid side chains. These side chains of certain amino acids can act as either acids, donate protons, or bases, accept protons. Acid-base interactions. So now we're talking about donation and acception not electrons, but protons, known as hydrogen plus. So acid-base interactions can play a role in stabilizing the three-dimensional structure of proteins. Here are some examples. So we have aspartate glutamate interactions, where aspartic acid, asped, and glutamic acid, glu, are acidic amino acids that can donate protons. They can form acid-base interactions with basic amino acids like arginine or lysine. Histidine has a side chain that can act as both an acid and a base depending on its protonation state. Interesting. It can form acid-base interactions with other amino acids, especially in the active site of enzymes where proton transfer is involved in catalysis. And we have cysteine, which has a thiol group, the SH, that can act as a weak acid. It can form acid-base interactions in the presence of other amino acids with basic side chains. Serine threonine interactions. Serine, SCR, with the OH, threonine also with the OH, but it's with the T, have hydroxyl groups that can act as weak acids. They can form acid-base interactions with amino acids, contributing to the stabilization of protein structure. Tyrosine, which has the OH at the end of the phenylalanine, has a phenolic hydroxyl group that can act as a weak acid. It can form acid-base interactions with other acidic or participate in with other amino acids or participate in hydrogen bonding with the protein structure within the protein structure. These are super important for the MCAT and we'll definitely get questions from them. So let's add these to here. So we have aspartate glutamate interactions. They can donate protons, they are acidic. Histidine is tricky, it can be both acid and a base. So bright. Oops. Active sites of enzymes where proton transfer is involved in catalysis. So active sites of enzymes. It's with histidine. Cysteine, weak acid, because it's our sister. Q, our sister. Weak. No misogyny though. Then we have serine, threonine. Weak acids. Stabilization of protein structure with serine and threonine because they have the can make acid base interactions. And lastly we have tyrosine, which is phenolic hydroxyl group that can act as weak acid. What's common between these weak acids? Cysteine, serine, threonine, tyrosine. It can hydrogen bonding within the protein structure with tyrosine. Okay, that should be enough. And let's put it is we can also flag these. So it's good to actually flag them. Denaturation is when a protein or nucleic acid loses its quaternary structure due to quaternary, tertiary, and secondary structures due to breaking of non-covalent bonds. This is important, not heat. I kept answering this as heat, but it is the breaking, the cleavage of non-covalent bonds. So non-covalent bonds are these hydrophobic bonds that happen between these functional groups within the tertiary structure, or even quaternary structure or anything else, right? So here we can see that for the tertiary structure we have the van der Waals bonds, the hydrophobic disulfide bridges, and hydrogen bonds. So, in 4 different ways you can denature the proteins. You can unfold them with temperature. You can't unfold the primary structure with temperature because it's an amide bond and it requires water, right? So with chemicals you can, with enzymes you can, with enzymes you are breaking down amino acids, and with pH you are able to break the acid base interactions that happen in ionic bonds, that happen in tertiary and quaternary structures. Are some of acid base interactions ionic bonds? Yes, some acid base are ionic bonds. So aspartate with a negatively charged side chain at physiological pH. Arginine is a basic amino acid with a positive charged side chain. Aspartic acid and arginine can form ionic interactions in proteins. Glutamic acid is negatively charged, lysine is positively charged. Glutamic acid and lysine can form in ionic interactions. These interactions contribute to overall stability and structure of proteins. In addition to ionic interactions, other types of acid-base interactions in proteins may involve proton transfer, hydrogen bonding, and interactions between ionizable groups in amino acid side chains. What are van der Waals forces? Not van der Waals. Are weak attractive forces that arise between molecules or atoms due to fluctuations in electron distribution. So we have London dispersion forces, LDF. We have dipole-dipole, hydrogen bonding. And interestingly, hydrogen bonding is a specific type of dipole-dipole interaction that occurs when a hydrogen atom is covalently bonded to a highly electronegative atom. Dipole-dipole is between molecules with permanent dipoles. So the hydrogen is the plus side of it, and the oxygen is the negative side. Oh, here we have examples now. No. What are examples of dipole, and LDF? So we have hydrogen fluoride. It's halogen bonds, right? Creating a permanent dipole. So the fluorine is the negative side. Chloroform is a polar molecule with a dipole moment due to the difference in electronegativity. So it's all coming down to electronegativity. Interestingly, we have also acetone between the positive hydrogen atoms and the partially negative oxygen atoms. And then we have LDF like methane. Okay, so now it comes down to uh, the fact that methane is a nonpolar molecule with no permanent dipole moment. However, these London dispersion forces are present due to temporary fluctuations in electron distribution, leading to temporary dipoles in neighboring methane molecules. And we have Cl2, diatomic gases, even. These are London dispersion forces. So this should plays a role in attracting one chlorine molecule to another and have no permanent dipole moment. These are identical atoms like O2, Cl2. Benzene is a nonpolar molecule with a symmetrical hexagonal ring structure. London dispersion forces contribute to the attractive forces between benzene molecules. So any almost interaction is London LDF, London dispersion force. And these are all subtypes of van der Waals, right? So van der Waals forces are LDF in general, then more specifically dipole-dipole, like hydrogen bonding. Hydrogen bonding is a specific subtype of dipole-dipole, and dipole-dipole like hydrogen fluorine, chloroform. These oxygen, hydrogen, negative and positive forces. So yeah, and we have also benzene here that is a LDF. So depending on their overall molecular geometry in the presence of polar bonds, these molecules can exhibit a combination of dipole-dipole and LDF. Hydrogen bonding is a subtype of dipole-dipole. Cool. So let's actually write it. 3 types of van der Waals forces. LDF in general, molecules. CH4, for example. What else did we have? Diatomic gases like Cl2 or O2. Anything else that was there? Like benzene. Then we have dipole. And then we have hydrogen bonding. Subtype of so for dipole we have acetone, hydrogen fluoride, chloroform, Should be good. What type of enzyme catalyzes cleavage with the addition of water? So there's a hydrolase uses water, and an example for that would be pyruvate dehydrogenase or phosphatase. Is pyruvate dehydrogenase a hydrolase? No, PDH is not hydrolase. It's oxidoreductase. Hydrolases are enzymes that catalyze hydrolysis reactions where a molecule is cleaved by the addition of water. PDH is involved in a different type of biochemical reaction. It's the conversion of pyruvate, a 3-carbon compound, into acetyl coenzyme A, CO2, and reduced nicotinamide adenine dinucleate, NADH dinucleotide. This reaction is a crucial step in the conversion of pyruvate, produced during glycolysis, into acetyl-CoA, which enters into citric acid cycle, Krebs cycle, for further energy production in the form of ATP. So we have to do this again because we missed the example. So phosphatase is what uses water to do that. What type of enzyme catalyzes cleavage without the addition of water or transferring of electrons? That's a lyase, not ligase, because that's the opposite, exact opposite, but lyase. An example for that would be, let's see, is it the one that turns G6P into F6P? Or is it the one that cuts down fats? That should be a lipase. So yeah, for this I think it's okay, this is pyruvate decarboxylase. It turns pyruvate into acetaldehyde, catalyzed lysis of a substrate, generating a double bond in a non-hydrolytic, non-oxidative elimination. Not oxidative or hydrolytic, so non-hydrolytic, non-oxidative elimination. And generating a double bond. The double bonds form here, as you can see. So pyruvate decarboxylase is a type of lyase. What are examples of lyases? Pyruvate decarboxylase is not here, but anyways, these carboxylases are all lyases. Carboxylation of PEP, for example, to form oxaloacetate. It's in gluconeogenesis. Isocitrates into succinate and glyoxylate in glycylate cycle of certain bacterium plants. Fumarase and citric acid cycle catalyze the reversible hydration and dehydration of fumarate to form malates. So they do this removal addition of groups from substances without hydrolysis. What are flavonoids? Is this what it's in rice? any of a large class of plant pigments having a structure based on or similar to that of flavone. Flavone is a colorless. It's a colorless crystalline compound which is based of a number of white or yellow plant pigments. Should be like rice, right? So let's see. Is flavone in rice? Rice, including both white and brown rice, contain flavonoids, but the specific types and concentrations can vary depending on the rice variety. They are in the outer layers of rice grains, particularly in the bran layer. Brown rice, which retains the bran and germ layers, tends to have higher levels of flavonoids compared to white rice, which has had the bran and germ removed during processing. The specific flavones present in rice can include compounds like apigenin, lutein, and others. These flavonoids contribute to the color, flavor, and nutritional profile of rice. Keep in mind that the overall flavonoid content in rice may be lower compared to certain fruits, vegetables, and other plant-based foods, but rice can still be a source of these beneficial compounds in the diet. What type of enzyme joins two large biomolecules? It's ligase, often of the same type that happen in DNA, right? Catalyzes interconversion of isomers, so it's isomerase. Now the example would be like the ones that we had, yeah, so G6P to F6P, right? So phosphoglucose isomerase, phosphoglucose. Why is phosphoglucose isomerase named as such? Explain it by relating the name to its function. It catalyzes the glycolytic pathway. So phospho indicates the substrate of the enzyme is phosphorylated. In this case, the substrate is glucose 6P. The glucose specifies the type of phosphorylated sugar involved. It is glucose in this case. And isomerase indicates the type of enzyme in reaction catalyzes. Isomerase converts one isomer into another. The context of phosphoglucose isomerase, it catalyzes the conversion of glucose 6-phosphate, G6P to F6P. So it succinctly describes the enzyme's function. Isomerase, which phosphorylates glucose, converting G6P to F6P. This reaction is a crucial step in the glycolytic pathway. So you break down sugars to produce energy in the form of ATP. So is fructose another type of monosaccharide? What's the difference between glucose and fructose? They're both simply sugars, monosaccharides, with the same molecular formula, but they have different structures. The structure of glucose is a straight chain with aldehyde functional group, but fructose is a ketone rather than an aldehyde. Hmm. Fructose is sweeter, so glucose is found its primary source of energy in human body, and under glycolysis or cellular respiration it's processed. It's less sweet and than fructose, and it's used in medical and sports beverages. That's where you have glucose, sugar, right? But fructose, it's in honey, certain vegetables, fruits, and it's associated with the sweetness of fruits. And so fructose is a keto. It's a ketone, and that's why the oxygen, the double bond, is not at the end. It's on the second last carbon. But for glucose, it's on the last carbon, which makes it an aldehyde. So that's why you have a phosphate, which adds a phosphate, right? It's phosphorylated. Oh, it indicates that the substrate is phosphorylated. So G6P into F6P. Wait a second. Wait, but G6P is not phosphorylated to turn into F6P, right? So I think the phosphate part, just because it's related to G6P that has phosphate and also F6P that has phosphate, so it's phosphoglucose isomerase. So the phosphorylation occurs before phosphoglucose isomerase, and it's by hexokinase or glucokinase, depending on the tissue. I think one is for liver, the other one is for the other cell membranes. So glucose plus ATP gives G6P, catalyzed by hexokinase or glucokinase, and then G6P turns into F6P. I appreciate your diligence in catching that error, and I hope this clarification is helpful. ChatGPT knows how to play with words. Okay, so now we have phosphoglucose isomerase down. We can do that again. This is the guanidinium group. So we have 3 of these different groups. I believe so. We have an indole group. We have a guanidinium group and another one. Okay, I think there's one that's with arginine. This is the one with arginine. So how I memorized this was that so I due to practicing a lot of the arginine with there's an app for amino acids that you can download on your iPhone and you can use that. I know this is arginine, right? It's the carbon attached to these 3 nitrogens. Somehow I related to Argentina, their football team or whatever. They like to play with 4-3-3 formation, and these are 3 nitrogens. I don't know if it makes sense for you, but this is how I prefer to memorize it. And then I related that to Guantanamo Bay, which is close to South America. So Argentina, Guantanamo, guanidinium. So should be close enough for now. So let's do that again. What type of enzyme adds a phosphate group from ATP to a substrate? It should be, so whatever is, whenever you're adding, it's a phosphatase. Sorry. Yeah, I mean, you're taking the phosphate and adding it somewhere else to a substrate. I mean, we have kinase adding phosphate, phosphatase taking away. It should be kinase adding phosphate. Yeah, so kinase brings the ATP and adds the phosphate to protein, minding its own business. Kinase ATP, and then the ADP goes off and the phosphate attaches to him. But is he more energized? What's the relationship with energy? I'm just being deep here. So, yeah, although I got it, I just put it hard. What occurs when two cysteine molecules are oxidized and create a covalent bond between their thiol groups? This is a disulfide bond. This forms cysteine, not cysteine. You just remove the E from the middle. I knew that. So this is a cysteine. You can call it cysteine, just to be safe here. Cysteine chapel. What is that? Cysteine chapel. Chanel. Combining two sulfurous eggs. Okay. What removes a phosphate group? It's phosphatase. And we know that phosphatase is a type of lyase. And these are all hand in hand together. So the stereochemistry of the alpha carbon in all eukaryotic amino acids is L, amino acids. So L is the stereochemistry of the alpha, except cysteine, which is L and S, R, which is R. Lysine, S, R, L or D, LD. Does that make cysteine a D? So does L-cysteine make it a D amino acid based on the stereochemistry of the alpha carbon? No, the L-cysteine as in amino acid refers to the stereochemistry, not the configuration of the sulfur. The L is on the left side, carboxyl on the right side. The hydrogen atom is pointing toward U, so L-cysteine and L-amino acid based on the stereochemistry around alpha carbon. The thing is, with all of these, this one, so this is number 1, this is number 4, this one is 2, and you go like this, and you reverse it because you're passing through the hydrogen, so you get the S, not clockwise, but anti-clockwise. For cysteine, because you're going first from here, because S is in higher priority than oxygen. You go this way, and then when you reverse it, because you're passing through the hydrogen, you get an R. So that's why L-cysteine is an R configuration. still it makes it an L-cysteine. So all the amino acids are amino acids. On the alpha carbon, their stereochemistry is L. Only glycine, because it has hydrogen, is achiral. That's it. What enzyme adds a phosphate group from an inorganic phosphate like HPO4 to a substrate? From an inorganic phosphate, PI, like HPO4 to a substrate. So you're adding PI, and PI plus ADP gives off ATP from the reaction. This should not be. Is this kinase adding a phosphate group? No, it's a phosphorylase. So we have phosphatase and phosphorylase. And this can be very tricky since we think that it is cutting down a phosphate group, but phosphorylase is actually adding an inorganic phosphate to the molecule. Okay, phosphorylase. This is very important. What structure of proteins is the interaction between peptides and proteins that contain multiple subunits? Multiple subunits, it's quaternary structure. This is the interaction between peptides, alpha subunit, beta subunits of hemoglobin. Yeah, I'll do this. The Michaelis or maybe I'll just do this. The Michaelis-Menten curve is hyperbolic. Yes. It reminds you of myoglobin too. What are used by the immune system to target a specific antigen, which may be a protein on the surface of a pathogen or a toxin? To target a specific antigen. Wait a second. Where are we? Which chapter is this from? This should not be 1 or 2, right? But however, it's related still to proteins, so it might be chapter 1. By the immune system to target specific antigen, which may be a protein on the surface of a pathogen. Is this ligand? Antibodies. They're also known as immunoglobulins. Igs, these are antibodies. Lymphocytes after they find a pathogen. I'm about to end this man's whole career. This is a funny meme. I like that one. Major families of cell adhesion receptors. Cell adhesion receptors. Cadherins, selectins, Ig superfamily, integrins. Which may be a protein on the surface of a pathogen or a toxin. So let's now put this as again. What is the substrate concentration that gives you a reaction rate that is halfway to Vmax? It should be Km. Half of Vmax. This amino acid, it should be serine, SER, and S, right? Yes. Polar, and is it aliphatic? Yes. It's uncharged. Yes. I'm good with this too. What is the maximum rate at which an enzyme can catalyze a reaction? Maximum rate is Vmax. This is when all enzyme active sites are saturated with substrate. So we can ask, when does this happen? When does it happen? When does it happen? When all substrates, when all active sites are full with substrates. What has a rigid cyclic structure, which amino acid and can interrupt secondary protein structure is proline. It's kinky. It's rigid too. Proline and glycine. Okay, just give me a second and I'll be right back.